So I'll be actually talking about approaches that are complementary to what Anne just talked about. And I'm a bit nervous because I will be presenting some results from her paper. So we'll see, but with a different view. Okay, so first, why me? Why am I here? So I did my undergrad in microbiology and then computer science. So I've been working at the interface between the two for quite a few years. So I think it's interesting to be able to read the very hardcore machine learning literature and think about how it fits or not fits the biology, and the other way around, look at the experimental setup and see how it fits or not fit what the ML is doing. So what's omics? So now we move out of the image field and we look at different ways in which we can make measures on cells. So omics emerged in the mid-90s with microarrays and shotgun sequencing by cellular genomics, for example, and the introduction of proteome-wide mass spectroscopy. So these were all techniques in the lab that were suddenly giving out a lot of data on different samples. And I'll put a few numbers out there for the more machine learning inclined in the room. Typically, we would get maybe between 1,000 and 1 million measure that we can get on a single sample, depending on the different omics that we use. So that would be the dimensionality of the input. And then the number of samples that we can assay is very variable. So it goes between a few dozens to up to maybe 10,000, 20,000 for consortia wide screening. So you get an idea that we're in a low data kind of situation most of the time. This data look into a lot of different types of molecules. For example, genomics look at DNA, transcriptomics looks at mRNA, proteomics look at protein, and then there's a whole slew of others like metagenomics, metabolomics, and epigenomics, and all sorts of other. I'd say that every month or 2 months, there's a new omics that come in, okay, to give you an idea of how it works. So the main important thing about this type of technique is that they try to give you a more holistic view of what's going on in the cell. Instead of just measuring one aspect of the phenotype, they really try to look at everything in their domain. So today, the presentation will be oriented toward predicting small molecule properties using omics data. What properties? For example, response in a screening assay, predict response in a screening assay, trying to find active compound or toxic compound that you want to avoid. The technique that I will show have also been used for mechanism of action identification and target identification. I won't go into these details today. One aspect that is really, really important is that everything I will talk about will work only on compounds that are already synthesized. So everything you've heard in the course so far on huge pace of 10 at the 60, at power 60, we cannot do it here because we need the data in order to make some kind of prediction. So we're dealing with much more confined molecule space. Finally, these techniques have been used to deal with drug repurposing and synergies. Anna has been talking about using drug repurposing when you're screening for compounds that are already drug and you want to use them in another indication. And ideally, we want to use multiple modality to make these predictions. So we'll go into that aspect a lot more. So what do we do? So what you've seen so far, you take molecule, a basic representation, most of the time a graph, or maybe you've discussed the use of fingerprint on molecular structure, and then you would make some measure on those, which can be computational, and then you try to predict a property. Sometimes you go directly from the basic representation to the predicted property, and then it's just standard form of gradient descent in order to optimize these different functions. So today I will focus on a situation where on the molecule, you actually don't work from the graph, you work from the powder. And then you make some experimental measurement on those, typically cell-based, in order to predict the properties. So we'll really focus on that specific domain of taking the outcome of an experiment on the compound and using this to predict something else. So there's a lot of data generation technique that comes into omics. So I've tried to layer them in different types of molecule that they query. How do I point? Okay. So the one that is working with DNA, that is the most frequently used one, would be whole genome sequencing, WGS. We won't get into that today. The second one would be bulk RNA sequencing, in which you quantify how much of each mRNA is present in the cell. And you end up with a nice vector of real numbers. This is really easy to get into all classical machine learning algorithm. Parallel to this, there's the L1000 experiment, which is slightly different. It measures only 1000 gene, but is much cheaper to run than the RNA-seq. Finally, I'll touch a bit about single-cell RNA-seq also. There's a lot of other that would have been interesting to touch using mass spectrometry. One that you might want to look into is called BioID, because that's one of the methods in which once you have a compound that is active, then you can figure out what protein is it touching. And in that way, try to recover what is the real target of your compound. So the data sources. So I know that machine learning people like to get their hand on lots of data. So there are nice database that you can use right away. The size of these data sets are rather small, as I said earlier, ranging between 1,000 and a few tens of thousand example. So you are in low size of data set for deep learning standard. One thing you have to know, if you come from a more computer scientific background, is that those data sets are very costly to assemble. Okay, biology is messy, so they are noisy also. So as a computer scientist, the biggest mistake that you can do when you start collaborating with biologists is start criticizing the data. Don't do that. Don't do that. Okay, they did their best, and it's the best that can be obtained. So there are two different types of data you will obtain there. There's going to be patient-derived data, and there's going to be cell line-derived data. So let's go a bit more in the patient-derived data. These are interesting because they come from real human beings with their genetic background and everything. They are very close to the disease because most of the time the cells are picked from the actual cell that caused the disease. So these are the most interesting data set to work with. These are the most messy also. There's the few one that I can mention would be UK Biobank, 1000 Genome Cartagena here in Quebec, that are SNP array based or whole genome sequencing based. So these are really looking at the DNA content of those cells. And then there are the transcriptomic data set, which I will talk a bit more later, which are the TCGA and the GTEC data set. TCGA is for cancer cells in patient, and GTEC is for normal cell in patient. The other thing I have to say on patient-derived data is that it's not directly available on the internet. Okay, most of that data that is very interesting to work on, you have to jump through the hoops of getting access to the data. Don't fear that part, okay? I feared for a lot of time, but now it's really simple, okay? You just have to describe the project, how you will use the data, and wait for the approval, which is relatively fast, okay? And every time I did that, they accept methodology development as a proper use of that data. So don't fear that. Finally, there are cell lines derived data. So cell lines, as Anne said, most of them are derived from tumor cell. But on top of that, you have to know that these are specific tumor cells that grow in a plate. This is very peculiar, even for a cancer cell. Most of the cancer would not grow on a plate. So when you work with cell line derived data, realize that these are very, very special cells, that they do not represent what's happening in the biology. On the other hand, they are super stable. The same cell line in different lab would behave roughly the same. So from a data perspective, they are much more reliable than patient-derived cell. In those cases, there's no ethical approval to get that data, so you can just download it from the web or from the different database and then use it. Okay, one small parenthesis here. Well, first, if you have question, don't hesitate to interrupt me. Okay, it's better so that we understand as we go. One thing I have to say is you've seen lots of very fancy machine learning approaches, but in biology, it's very annoying when you want to develop those methods to see that simple linear methods work sometimes very, very well. Okay, so before attempting anything fancy, start with the basic and then move up, showing that the complexity that you introduce in the analysis actually gives you something more. The reason for that is quite simple. Genes are correlated between themselves. So it means that if you have 20,000 dimensions for each of the 20,000 genes, they lie on a lower dimensional manifold because they are correlated. So they are not independent from one another. So by using that, you can just recombine those and find measures that are very interesting. So you have to deal with that. One of the reasons why you will see in lots of bioinformatics paper doing machine learning, the first thing they do is they do a PCA, reducing to 500, 1,000 dimension or so. And usually that gets rid of lots of those linear correlation between genes. So go for it, do that. It's a good idea. It gets rid of some of the noise also. So now let's get into multimodal omics. So we've seen there's different way in which you can capture these type of large profile on different types of sample. Now it would be nice to use them side by side. For example, using images side by side with mRNA profiling, side by side with proteomic profiling, or with the structure of the compound. Why not? Okay. There, I will introduce two concepts that I think are super important. The first one, and computer scientists are not easily grasping this part, is the fact that biological systems are infinitely dimensional. So there's absolutely no limit with the quantity of feature you could add through new experiment or just by going lower in the type of data processing that you apply. So you have to deal with that. Anyway, you will not have the complete data. That's for sure. You're always missing something. You have to choose what you're missing and deal with it. The second part is that multimodality, there is no systematic approach to combine data from these different modalities. If you look at the literature, it's a complete mess. Everybody does completely different things in different ways. There's no benchmark, there's no metric. So every new paper introduces a new metric and a new method. So you can't really compare between different approaches. You will see the consequence of that is that every paper find out that their method is definitely better than all the other ones. And if you reproduce it, if you try to reproduce it, it's going to work. Okay, it's true, but they add their own metric most of the time. There's a very serious confusion regarding what data level you want to use. For example, in RNA-seq, the lowest level that you can have is actually short sequences that are obtained from mRNA that are just sampling the molecules that are in the cell. So you could stay at that level and you would get maybe, I don't know, 30 million of those sequences for one given sample with all their sequence. It's nice. But then you can combine them saying, I'm just going to count how many reads appear on that gene, and I will turn that into a real value. So it's nice, but you lost something by going up in the processing. What did you lose? You lost splicing, how different part of the gene can recombine to can get stitched together. So this is lost. You lose mutation that you could see on the sequence of the gene. So in any case, you always decide to remove some information. Finally, when you work with biologists or computational biologists, you will hear the word normalization a lot. Biologists like their normalization. We've seen one with batch effect. So in that situation, you have different impact on your data set from different batches. And when you normalize for that, you want to remove that information from the data set before proceeding. Normalization is about destroying some information in the data set. So anytime you use data that has been normalized, try to think if the normalization, the information that has been destroyed by the information, will it be important for you? Does your method can deal with it? If so, maybe you want to have access to non-normalized data set. So I like this paper. It's an old one, 2005, okay? So this is before high throughput sequencing and transcriptomic and most of the omics. But David Searle at that time looked into how will we combine different modalities and what are the type of approach that we can do. He derived two approaches that I think are the main one. So the first one is called the low-level integration, in which you would take the raw data or at the level of processing that you decided to use, and you would combine them usually through juxtaposition of the two arrays, and then you build your method to integrate those two data sets during the application of the method. That's a nice one. The one that is used most often is that you will take your initial data, you will process it to death. Okay, and then you obtain the final set of things, and then you combine those results from well-known methods for the two modalities, and then you combine them to use them together to make a prediction. So these were called in those days high-level integration versus low-level integration. Today we use the term late fusion versus early fusion. These are exactly the same thing. This looks simple, but in fact, you can think of a lot of different network that would do early or late fusion, but most of the time, some kind of intermediary between the two. There would be some part of pre-treatment on the raw data, and then afterward you integrate, and then you do further on the juxtaposition of the data. And you've got a few examples on this slide. And they are just variation of where you decide to start merging the data. You have to think about what you extract from the data, and once you merge, what you're losing. If you have a neural network, you juxtapose the two input, then whatever is on top lose the information of which feature belong to which modality. So you have to make sure that this is the right point to do that. What I find really interesting are methods that go in a very different way. For example, this one here, you would have one modality, a second, and a third modality, and then you do some kind of processing to end up with a lower dimensionality representation. And then you want to just reconstruct from that representation back to the input. So a sort of multimodal autoencoder kind of thing, or VAE are used in this context also. But then you don't really care about the low dimensionality. You will pick one of the previous one, which is a sort of synthesis of the different modalities. And you would take that as representative of the object you're working with, the cell line or the single cell from the patient or the patient-derived genomics. And then you would work on that. So in some way here, you do a sort of pre-treatment of your modalities that is completely independent with the task you will apply. And then you just extract those vectors and then you work with it. They are pre-fused before you start processing them. Single cell, low level integration. That's really interesting because maybe in 2015 or so, 10 years ago, we started to introduce methods that were able to do transcriptomics, measuring the expression of each gene in a given cell, but to do it cell by cell in a sample. So usually these type of data sets, you would get maybe 5,000 cells from which you would get maybe 10,000 reads. So you have a very coarse expression profile. Yes.
Hi, I have a question on the previous slide. So when you say like so like all on the combining like the two modalities, so when you're not using like it's like a lower dimensional representation, do you mean that you'll use um
You mean this one here?
yeah, so if you're using like I guess like the original modality, is it that you're hoping that your model will learn the context of the other modality and transfer that to
You mean why don't we use the bottleneck to be the layer that we want to use for further processing? That's super. That's a really interesting question, and that shows the bias from the biologist perspective. The idea is that very often we want to reduce to 2 dimension for visualization. That's the only reason. But everybody knows that reducing to 2 dimension is a lot of compression of your data. So that's why you would want to use a higher dimensionality as a pre-compression approach, well, as a keeping more information. That's the idea.
Gotcha. Okay, thank you.
Yeah. But you're absolutely right. You could decide to have 50 dimension as the bottleneck, and that will work perfectly.
I also have a question. Maybe I'm getting a little bit ahead of you, but has anyone tried contrastive learning with these types of multimodal data sets? And if so, has that been successful or not?
Specifically for multimodal data set, I don't remember saying it, but it's really possible it's out there. I'm not sure what you would gain exactly. It might be out there. Yeah.
Is it possible to get the low dimensional representation if one of the modalities is missing or
Oh, excellent point. So in that case, there's no clean way to do it. So what you would need to do is just to decide to cut some part of the network, assuming it's not going to give out any signal, and assume that this is going to be understood by the upper part of the network as I don't have information. It's very difficult to distinguish between I don't have information and I have information, but it's the average of the input, or even worse, no genes are expressed. It's purely zero. Okay, so that's very different than not having. We'll see a method later on that can deal with that in a clean way, but it's definitely more complex. Good. So, okay. So I put this in quote, treat your data with respect, because it came when we were doing machine learning on some type of data, which was mRNA sequencing. And we had a nice conversation with a machine learning scientist who's Hugo La Rochelle. when he was at Twitter. And at some point he came in and looked at what we were doing with the data, and he said, "You're just not respecting your data. You're putting it in a form that doesn't make sense, and you're really losing a lot of information." So I took that and decided to apply it quite extensively. It means that when you receive the data, you will receive it in two types of format that I will discuss after, that I think are not the way in which you should think about the data. The best way to think about the data is really to have your different modalities independently measured and in what we call a long format, meaning that you have all the factors that you use in order to get a single measure. When you have this, you're much closer to what was the experiment that was done. For expression profiling, you would have, this is sample 1, this is gene 2, and this is the measure of the expression of that gene in that sample. In this way, you're very, very close to the data. When you will receive these data, always ask, how did you represent missing values? That's a tricky one, and it can really mess up machine learning pipeline afterward. Proteomics, for example, they tend to do all sorts of fancy stuff with the messy data just to make sure they give you a number. So in the end, in the data you receive, you will not see missing data, but you will see bizarre kind of distribution that will make you suspicious. Another situation in drug discovery, when they compute some value like the IC50, the potency of a compound, they will often replace this by a string saying it's larger than 10,000. They don't really know if it's larger than 10,000. They just decided that in this case, they cannot evaluate it. So in some way, it's a missing value that they know is just above the range. So don't replace greater than 10,000 by 10,000. That would be a big mistake. The two types of data that you will see will be presented as matrices or graphs. Biologists love their graphs and they love their matrices. The reason for that is not so interesting. Matrices, they can load them up in Excel and then play with it in all sorts of ways. That is very difficult to replicate most of the time. So that's the main reason here. And the second type of data you will receive is in the graph form. Well, where very often what we do is we do multimodal analysis with the graph by just throwing in different types of object, whether they are genes, cell, disease, compound, and then you would link them based on some similarity approach between those things. So it means that from a computer scientist perspective, all these nodes are very, very different kind of beast. And each of those edges actually represent different methods also. Working with just the graph, you have to be aware of that. The other, I think, most important mistake here is that you're discretizing continuous measures. Very often, if you look at the activity of one compound against one cell type, you'll get a real value, okay? If you decide that you put an edge or not put an edge, then you're discretizing this value. You've lost a lot of information. Were you just on the border or were you at the extreme? That is lost. Finally, you will be presented with data that is called annotation, gene function. They would say this gene has that function or this gene is in that pathway. Be careful with that. So first thing, these are well-curated database of information. That's nice. There's a lot of attention to do it properly. But on the other hand, the annotation that you see mean that someone at some point decided that this gene was in that pathway for a given experiment. What does it mean for other genes that are not in that pathway? It means nothing. Okay, every gene could be in the pathway you're looking at. So the absence of information, the absence of link between a function and a gene doesn't mean that they are not linked. Okay, so you have to be very careful about that. In the human genome, last time I checked, it's about 40% of the gene that have no function. So we believe they have, they're not found yet. The last thing I wanted to say with this is that a lot of these gene function are actually derived from large omics data set. Most of the time, the exact data set that you're working with in order to make prediction. So you see how sometime, for example, if you take a gene, look at its expression profile between different samples, try to predict the gene function. Well, the gene function of that gene was probably determined by this exact method. So you will reach 100% test accuracy, but it doesn't mean much. Okay, let's go into a bit more specific here. So this is one, I think, of the best paper to work with multimodal data set and trying to work with the integration. So this is one paper coming out from the Broad Institute that looked into making prediction of activity of compound, either working from the chemical structure, working from images from cell painting, or from gene expression from the L1000 experiment. And they try to assess what's the best modality, what are the good combination of those modality. They try early fusion, late fusion. They decided to look into activity by compiling 270 different activity measuring assays in order to make multitask prediction on these 270 different assays. So it's a great setup. Now, one thing that they come up with as a conclusion, and that's the conclusion I got from that paper, is that cell painting is the best. Really, it's the one that encapsulates the most amount of information in order to predict the outcome of a compound for a certain assay. Expression profile would be second, structure would be last. So that's already an interesting conclusion, because I'd say 90% of what is done in ML to predict activity is done starting with structure only. Okay, so it's the toughest problem to solve. Nice thing, it opens you to 10 at the 60 different molecules, because you can easily make prediction on compounds that are not synthesized yet. Okay, so that's a good thing. Regarding the comparison between cell painting and expression profile and eventually structure, we see that this hierarchy also fit the number of features that was fed into those networks. So cell painting gave the largest amount of feature at 1,500 or so, and then cell painting was done with the L1000, so it's 1,000 dimension that was fed in, and finally structure was shrunk down to 300 dimension after graph convolutional network and all those things, meaning that the actual capacity of the network for each of those modality varies. There's more capacity given to the cell painting, there's a bit less for the gene expression, and there's even less for the structure itself. So it's hard to say if this hierarchy derived from an actual use of the modality, or it was just based on the not dealing with the equal capacity of the different approach. Yes, there should be a mic coming in. Oh.
I have to give you an update from just a few weeks ago, because we were playing with this data set and a postdoc in the group discovered that actually a lot of the power of cell painting is coming simply from the cell viability, just like how many cells are there. So toxicity being the kind of primary output. So that's actually a little bit shocking. So yeah. I just wanted to offer I guess it's a caveat to this, where the image data is not necessarily providing this beautiful, rich morphology that I just finished describing. In this particular case, it seems like a fair number of these are, if you just take cell count alone, you can predict actually maybe better than some of these other modalities. So just an interesting find. I didn't want to leave the misimpression that it's the morphology that's super valuable here, because we're starting to think maybe that's not true.
Good. Thanks.
Could it be that cell painting does better this way because it's a more downstream phenotype towards it's closer to the disease phenotype itself. So actually, if you go from structure to expression profiles down to cell painting, kind of getting closer to the disease phenotype itself. Is that a
Yeah,
reasonable hypothesis?
That's a great hypothesis. And I think this is one of the value of assays like cell painting, which is really, really down the line into all the information processing that appears within the cell. But you may lose information along that way. You might have different perturbation that somehow converge to a similar morphology that you would lose. So gene expression would be slightly below that. You might have differences in the gene expression in order to recover the same morphology, and then you would get access to that. So I think it's the same concept that I said in the beginning. You have to decide where you cut along those lines. Good.
I just wonder that this ordering of cell painting expression profiles and structures last really depends on what you are measuring, right? What's your objective? This, for example, if we're trying to predict binding affinities, that wouldn't work at all. So can you comment on?
Sure. Great question. True. These 270 different assays were actually very different assays. Some of them were actually biochemistry based, really looking at one single binders. Some of them were on whole cell. Some of them were on bacteria, fungi, human cell. So there's a whole diversity of those assays that are there. In this particular paper, they didn't look at precisely what kind of assay were working fine and what kind were working less fine for the different modality, but that would be quite interesting. And I agree with you. In another context, experiment that we've done showed that using the structure for a very low level, let's say phenotype, like binding, the structure works fine. Okay, and actually the use of gene expression is just more opaque. Okay, it doesn't give that information easily. Good. Okay. On top of that, when we look at the result from which they derived that morphology was better than gene expression, better than chemical structure, like what we see here, they went a bit deeper and they look at the actual AUC they get from that specific modality across all the different assay, the 270 assay. And what you see here is kind of shocking. So I hope everyone knows about the AUC when you try to have a classification algorithm and then you see, does your score is a good predictor of the classification? So these are the AUC that we see, for example, in that case with structure, with gene expression, morphology, and then combination of those methods. So we'll look here in a situation where a significant portion of those predictions fall below 0.5 AUC. Now, does that stand well with you? Should it happen? Yes. Yeah, you could just flip it and get better. So it means that there's something going wrong in the optimization. Okay, so it means that somehow the network decided that it would completely disregard what's happening in that dimension because maybe it's doing better on others. So that's one possibility. The other thing that we see is that for a lot of the combination, or a lot of assay in those combinations, you would reach 1 of AUC. So perfect prediction. That's a bit suspicious. Okay, maybe we should look into those. So the main point that I wanted you to get from that specific paper is you have to understand where it works, where it doesn't work, and why. And very often when you do that, you will get sort of a bad surprise. You will realize that the network is actually picking up on things that you don't want it to pick up. And that's a very, very important part because in this sense, it gets you closer to how to analyze the data for your purpose. So now we'll switch gear a bit and I will introduce some of the data set that we have started working with in the lab and that has led us to try to attempt new methods. So this data set is coming from actually taking patient sample of leukemia. It's nice because these cells can grow well in a lab environment and we can throw compound over them and look if they die. So that's a very easy phenotype to obtain. So we're able to do 5,000 compound against 20 different samples directly from patient cell. So we have a sort of different screen. We've got 20 different screens for those 5,000 compound. And then we realized that some of the compound would get activity in the same sample, meaning that those compounds are killing the exact same subset of cell. So from there, we can make the hypothesis that these compounds are likely targeting the same function, mechanism, pathway. Okay, they act in a similar way. We found that those compounds are not similar in structure at all. So that's quite interesting because when we go back to chemists saying, we think we have a compound that could work against these subtype of leukemia, then they've got a whole set of compound that we believe is acting in the same way. They love that because they can pick the one they like, the one for which the chemistry would be easier to work with. So we've got this nice approach. In the paper, we use it to predict synergy between compounds. So that's one thing I won't go into. But then for those samples that we have assayed, we also had the RNA-seq. So we were in a situation where we did have those two different modalities, compound screening and mRNA sequencing on the other side. And then we started to try to figure out if we have a given sample for which we have the chemical response, the chemical profile, and we have the expression profile, how do we make a prediction for that sample? We could fuse the two modalities and go up. We tried it. It doesn't work well. We were trying to predict prognosis, whether the patient will survive or not the standard treatment. And this was not working well at all. So the approach here came out from a discussion we had quite some time ago when we were discussing with Yeshua regarding how can we combine information that is not complete on both sides. There's missing data in this. They happen to be very different types of measures. How can we scale up our analysis? And one thing he suggested at that time was the use of embedding with a sort of reverse architecture network in which, for example, for gene expression data, you would feed, this is the gene name, or this is the gene identity. You would say, this is the patient identity, and you will train a neural network to predict what was the expression of that gene in that patient cells. Once you've done that, you train the embedding, the first layer, and you always can see the embedding as one hot as input into a dense layer, and then you just extract this dense layer. And that dense layer would be characterizing each gene by an embedding, each sample by an embedding, but also each compound by an embedding, because you train one in parallel to predict the chemical response. So essentially what you're asking by doing this co-training, usually by alternating batches between the two modalities, you're forcing the network to combine in one sample embedding the information to reconstruct the chemical profile and the expression profile. If you're missing some gene, no big deal. You just don't present them to the network. If for some sample you don't have the chemical profile, no big deal. You just do not present this information to the network. So we thought that was quite interesting, and then we started to work with it. We found out very rapidly that the data was really, really not large enough to do any of such fancy thing. So we sort of abandoned the project. But we kept with the idea of working with embeddings in order to condense information into a more palatable form. I'll go through three I'll go through rapidly through 3 papers that introduce different type of different angle. So the first one that came out was called Gene2Vec. So maybe some of you have heard about Word2Vec in natural language processing, you know, that this is everywhere, replacing a word by an embedding and processing it. So these other had the same idea. We would put an embedding for each gene, and this is going to be encapsulating all the information we have about that gene. We don't know if it works, okay? Because there was no metric to evaluate the quality of the embedding of a gene at that point. What they use is try to predict from those embedding whether those 2 genes would have expression profile across patients that were highly similar. That's a rather easy task. I'm not sure if it's really aligned with what we want to do with those embeddings, but anyway, this is what they report. When we look into the paper again looking at the details, we found something surprising. We found that in order to train their embedding, they go through 10 epochs, only 10 epochs. Absolutely no discussion of whether the network converged or not. They just leave it at that. On top of that, they decided to pick the epoch based on the validation data set. So essentially, they were cherry picking their epoch. I'm not sure why you would want to do that. Maybe to protect yourself against overfitting. Maybe that would be the reason. But then first show overfitting, show training versus validation metrics, and show that it's actually going up. So they don't do that. They didn't. So they went through 4 different architecture of the network, and then they picked the best. If you look at the numbers in those tables, you will see that epoch by epoch, the clusterness, which is the quality metric that they try to use, goes up and down through those epochs. So this is a very good suggestion that either the whole thing is really noisy and unstable, or that it hasn't really started to learn properly. You would need to go way farther down the road to make it at least stable. When you look at the accuracy for different architecture, it doesn't seem to follow much trend. So they are pretty much the same, but slightly up and down at different epochs. So we're not sure if they picked the right one. And then the other result that they show is that when they train their embedding on expression profile and then tested them into their interaction between 2 genes, they try 2 things. First, fixing the embedding in learning their task, or allowing for fine-tuning of their embedding on their specific task. When they do fine-tune, they reduce their accuracy, which is always a bit suspicious. Could happen through overfitting. That's a great hypothesis, but then you have to show it just to make sure it's that. It might be something else. So one other paper that is really going a bit further is the paper by Choi et al., in which what they decided to do is to give an embedding to a gene and give an embedding to a sample, and then they do a dot product of these embedding, and that should be predicting the expression of that gene in that sample. Why did they choose? Why did they choose dot product? Not sure. Seems to work a bit in their system, but let's think about what impact does it have in those embedding. Using the dot product as the fusion function essentially means that both your gene and your sample must reside in an equal dimensionality space. Is it something that you want? Well, not really. These are really 2 different things. Okay, and the other aspect is that by just doing a dot product, you're essentially saying are the 2 vectors in a similar direction. When you do that, it means that your actual Euclidean space in which you find those embedding has to have the same semantic If you take one portion of the space in which a gene lie, it has to also have the sample for which that gene is going to be highly or lowly expressed. So in some way, you're adding a lot of constraint on your approach. I won't go into the detail just to make sure that I present the other method. And the method that we've presented on that with Asya Trofimov, which is now looking for a faculty position, if someone has a good idea. The idea was that we would take embedding for sample and embedding for genes, and we will essentially combine them through a deep neural network. 5-layer, relatively small, to deal with the rather low data set that we had. Essentially allowing the two embedding to be in different space that are absolutely not linked with one another, because it's a complex nonlinear method that makes the link between the two. So each embedding is completely free to have its own semantic in space. We trained the network for 500 epochs in order to see some sort of convergence. But at the time of writing this paper, these 500 epochs were taking 72 hours to train, which is a bit annoying for the kind of resources we had. We found out that this was a simple bug in the way we load data to the GPU that made it so slow. And now we're training it in 2 hours. So meaning it's a rather fast method, we could go to 5,000 epochs very easily. We found that the quality of the embeddings that we get at 500 epochs is much better than what we've seen with the other method. And now recently we're training for 100,000 epochs, and we see that the results are really, really, really better when you go farther down the line. So this makes me a bit suspicious on the two previous methods that were training respectively with 3 epoch, and the other one was training for 10 epoch. That seems to be a little on the low side. And then you might want to look at the paper to see the fine detail of the method. But what we found is that if we take a very simplistic task, which is given an expression profile, predict what type of cancer does this sample come from, then we see that the different types of embedding, the Choi embedding or our embedding or TSNE on top of PCA, ours get better accuracy in predicting the cancer type. We don't say this is interesting. Predicting the cancer type, you know from where you picked it, on which patient. But the idea is that the embedding is conserving more information about what's going on in the patient. That's the idea. So take-home message for today. Biology equal infinite number of input dimension. You have to decide where to stop. And machine learning methods are not really good when the dimensionality of the input space gets to infinity. Okay, that's never a good idea. The second thing is using multimodal approach in omics data usage. Despite it has been worked on for 20 years, it's still really, really not understood how to do it properly and what are the consequences of some of the decision. So I really, again, implore the people doing machine learning to get to look into these type of data set and bring your expertise on it. The current direct application of multimodal omics and drug discovery are still few. We're sort of getting there, but it's really, really just started. And finally, using the factorized embedding offer good opportunity to do omics integration. That's not something we've tried so far, but that's something that we plan to try very, very early on. And thanks for your attention.
We might have time for 1 or 2 questions before the break.
Hi, I've been working with a lot of the gene embeddings and multimodal data recently, and I was curious, the papers you're referring to are around 2020. I know that you've been working in this space for a long time as per your presentation. I was curious, in the state we're in now, where would I go to find more about like methodologies that are proper for it? Thanks very much.
It's difficult to figure out. I think the good papers are probably in the bioarchive or the archive. Surprisingly, these are kind of papers that are not trivial to get published for all sorts of reasons, mostly trying to find reviewers that really understand the intricacy of both sides. That's kind of difficult. And, you know, mining the archive is kind of an art that I'm not so good at, but I haven't heard of anything that really surpasses what has been done so far. You know, and that's a pity. Good. Well,
So a lot of the machine learning architectures have origins in the vision space and have like they are originally developed for computer vision. And my question was, what are some inherent differences that we should be paying attention to before using some of them?
Yeah, dealing with omics data. So they are very different in nature. They are very, very different in nature. One of the main features that are used to analyze image is proximity of those pixels. There's an inherent structure in an image where the pixels are not independent from one another. They mean something locally. That's something we don't have in the omics. So when you present a vector of expression for an mRNA sequencing, the adjacent gene are there for absolutely no reason. Actually, you would be surprised to see that most of them are ordered by alphabetic order of the name of the gene. So there's absolutely no information. If you try to apply convolutional neural net on an expression profile, you know, that's kind of wrong. If it works, you have to be really curious about what's happening, dealing with genes, starting with the same letter, contributing the same thing to the output, you know? So that's one of the main differences. But it's completely different when you do cell painting. There, the image analysis is really bringing you something, that's for sure.